Hello, so today we're going to talk a bit about DNA and protein synthesis and how they work together. Behind me I have my cell. So in the center over here is our nucleus. It is surrounded with our RER, our rough endoplasmic reticulum, which is studded with ribosomes, and then you also have your different little ribosomes all throughout your cell. This big red one right here is just a bigger, exaggerated version of a ribosome, so that way we know, it just makes things easier in how we can explain it. Also in our cell we have our tRNAs, so that's your transfer RNA, and that has an amino acid attached to it, which will help build your proteins. So to talk a little bit more about the DNA. So your DNA is a double helix. It is composed of a phosphate sugar backbone. So your template strand will start off with the sugar, phosphate, and a complementary strand would be phosphate, sugar. And all of these little bonds together are your nucleotide bases. So to start off, you'd have your adenine, your thymine, your cytosine, and your guanine. And those, your A always bonds to your T, it's a double bond, and then you have your C, which binds to G, which is your cytosine to your guanine, and that is a triple bond. So they can only bond together because of how many bonds there are. So with your DNA, so it would be twisted together, and then DNA helicase would come in, and it untwists it and opens it up. As that happens, your RNA polymerase will come in and it starts reading your template strand. As it's reading your template strand, it's making a copy which would start to come out through and give you your mRNA. Now with RNA, you don't have thymine. Your thymine gets replaced with uracil. So all these little white dots will represent your uracil. So your mRNA, it goes out through your nuclear pores out of the envelope, and it goes into, and it finds a ribosome, and then that's where it starts to make protein synthesis. So your protein synthesis, your ribosome your messenger RNA comes in, and it starts to read in through the ribosome. The ribosome clams shut down on top of your RNA, your mRNA. So as it's going, it reads it in sets of 3s, so you have your codons. So your start codon would be your AUG, and you would have a bunch of different stuff. Before your AUG, your start codon, you'll have a bunch of other junk at the end of it, after your stop codon. So as your mRNA starts to feed in through here, once it gets to your AUG, so it'll read it in 3, your AUG codon gets read, and when that happens, where did your AUG go? So your AUG comes in, and its anticodon at that point would be UAC. So your UAC, your tRNA comes over. It has a amino acid attached to it. Once it reads the AUG, it plugs into it, and it drops off your amino acid. So that your RNA, your tRNA, then goes back into the cell and hangs out and grabs another amino acid ready to go, and it keeps going back and forth. So you've got your amino acid plugged in on, and it gets set into your ribosome. So your mRNA still gets read through your ribosome, and as it gets read, and you'll have different other codons that will come in. So this is your GAC. So your GAC comes in, plugs in, and it drops off its amino acid. So your amino acid gets dropped off. Your tRNA then goes back into the cytoplasm, and this continues on and your amino acids will just continue to come in and drop off all of the different, your tRNAs will come in and continue to do the same process. So once you get the stop codon that reads through here, your RNA falls off, and that actually will go and get recycled inside of the cytoplasm. So when you hit the stop codon, all of your amino acids then will fold and create their protein. The proteins are essential because they build everything in your body. So all of your muscle groups, every cellular structure in your body is composed of proteins. So each unique protein has a different set of amino acids that are required to build that. So it folds, you have your protein, and then that goes off into the cell wherever it needs to be made. And that is protein synthesis.